Since 1977, the Saba Lions Club has been a pillar of service and community care on the island. What began with 20 committed members has grown into a legacy of giving back, supporting those in need, and advancing the global mission of Lions Club International. From cultural events to signature service projects like Meals on Wheels and the Food Bank, the Saba Lions Club supports the elderly, disabled, and struggling families with compassion and consistency. Their health programs, like annual eye care support and glucose fairs, continue to promote wellness across the island. The club is also a champion for the environment, proudly maintaining an adopted hiking trail and backing local cleanup efforts to protect Saba's natural beauty. Several of its members have been honored with the prestigious Melvin Jones Fellow Award for their outstanding dedication, proof that the spirit of service is alive and well on Saba. The Saba Lions Club, serving with heart for the good of all.
Welcome to See and Learn, everyone, to our second last presentation of the month. For those of you who are tuning in by live stream, we'd like to thank the Lions Club. They are the reason we are able to live stream this presentation tonight, along with Chizilala Productions. So thank you, Lions Club. We'd also like to thank our major sponsors, Public Entity Saba, the Culture Funds of the Caribbean, and the Nature and Environment Policy Plan. So big round of applause for them. Without them, this program would not be possible. And a big thank you to all of our sponsors. We have almost 60 sponsors across the island, on St. Martin, and internationally. So thank you to all of our sponsors. And tonight's expert is sponsored by Althea's Cottage by Jeff. So we'd like to give a big shout out to Jeff. He's a big, important sponsor for us. Raffle tickets. Several of you have bought some tickets tonight. If you have not, we are running out of time. Our goal is to raise $5,000 to support our Critter Quest scientists in 2026. So please, it's $5 a ticket. We have over $8,000 worth of prizes, including a liveaboard, hotel stays, jewelry. What else, Lynn? Flamboyant Cottage, 3 nights at Flamboyant Cottage, or more, if you want to pay some more. Handmade summer morning, and a bunch of other things. Yeah, 1 week on St. Croix, a stay in St. Croix. So lots of excellent prizes. So please consider supporting our raffle. As we're winding down in October, we only have 2 more See and Learn events. Tomorrow afternoon, we have a dive with Elwin and his team. Luke, I believe, is in the crowd, so raise your hand, Luke. If you're interested in diving with Luke and learning more about whelks and the restoration efforts the Conservation Foundation is doing, please sign up with us. And then our final presentation and closing night on Monday will be at Amanhana with Elwin. So we hope to see you there. This clicker. And we have one more field project. If you have not yet collected your hat, or if you want to go out on the dive tomorrow, you can stop by, pick up your bucket hat or baseball cap, and they're made from recycled materials. They're very nice. So if you have not collected it yet, please see us. So let's filter through the facts with Dr. Aaron Pilnick. What began with backyard reef tanks in New York has evolved into a career dedicated to coral and sponge restoration across the Gulf and New England. After helping start the Florida Aquarium's coral restoration program, Aaron earned his PhD from the University of Florida, studying restoration aquaculture and marine invertebrates. Now a postdoc at UF, he's building the framework, spicule by spicule, for stronger coral and sponge restoration. Please welcome back Dr. Aaron Pilnick.
Cool, thank you. Is this working? Is my mic working? Whoa! So, yeah, I'm going to try. My goal for tonight will not be to fall in the moat. So, and if I do, then that's my fault. So, and I'll just work through that. But yeah, thank you to Emily, thank you to Lynn, thanks to See and Learn for inviting me back. I was here 2 years ago, and it's just an amazing experience to be able to share our science. I mean, this is really in paradise in the tropics, and to be able to interact with everybody here who has a genuine interest in the work that we're doing. So. It's really special. The program's really, really quite special. Also, thank you to Alwyn and his team for hosting us in the research lab for basically this entire week. So yeah, we'll get started here. So, all right. Well, before I jump into the topic here, I just want to kind of really briefly give a teaser here. So, about a separate but related project that we're working on. So 2 years ago, when I was here, one of the school activities that I did was we went actually into the field, into the reef, underwater, and we took some VR, 360-degree footage, loaded it onto these headsets for these kids in the school, and actually was able to show them what it looked like to be on the reef. And these are for kids who are a little bit too young to be snorkeling or scuba diving. And so this was a really cool opportunity to kind of share, what their backyard looks like, right? But without actually having them get into the water. And I think that was a pretty good success. And so we're actually back again this time with my brother, Alex. Hey. And so here he is on the left here with the 360-degree camera, and this is a footage from Man O' War Shoals. This is a really quick preview. We did this activity again with the kids, but the idea is for us to develop these educational experiences for the kids that'll be a little bit more interactive. It'll be a little bit more of a formal learning experience where they'll be able to actually answer some multiple choice questions, look at sponges, and so we're really excited to roll that out with See and Learn. And so thank you guys also for that project. All right, so this was kind of, when folks were asking me, what's your topic? And I said sponges. It's like, oh, another sponge talk. And I'm like, okay, well, I promise this one will be a different type of sponge talk. Yeah, I think it's really quite interesting. There's quite a good sponge diversity, as we've been seeing in our dives this week. And today I'm really going to focus more on the restoration aspect. So I hope most folks here were at Don's talk a couple of days ago, and we'll kind of get into some of that. But he gave a really good primer for the work that I'm going to talk about now. But seeing as we're kind of talking about restoration, I thought I'd give a little bit of background on myself and kind of why I'm doing this, right? Why I'm interested. And for me, it really all started, apart from keeping aquariums in our dad's basement in New York. In undergrad and college, I had a pretty foundational course in tropical ecology in Costa Rica. And for me, this was the moment I realized that I needed to study ecology, right? And then at the same time, the Deepwater Horizon spill happened, right? And so imagine you're, a young person learning about science and biology, and then this environmental tragedy happens. And so I've really been focused on, for a lot of my career so far, trying to understand how do we improve these coastal habitats, right? How do we try to reverse the course and try and restore some of these ecosystems and hopefully leave them in a better place than we found them. And through that work, I've worked a lot with Alan and his team here on sea urchin aquaculture, and you'll hear an excellent talk from him on tomorrow, I think. Is it on Monday? And so this has been a really great partnership. We've been working with Alan and his team since I think 2020. And so that work is still developing, and I'm really excited to tune into his talk on Monday as well. But back to sponges. We're going to talk about sponges again. So here's a picture of Don in front of this really amazing big barrel sponge. These are the redwoods of the Caribbean, and divers here. For me, looking at these big barrel sponges is really, really, really amazing. And there's such a diversity of sponges here. There's these branching purple tube sponges, the barrel sponge, and if you were to look around in the headset, you would see probably at least 12 other species. These reefs are really, really diverse and filled with sponges, and so we're going to kind of talk about that. If for anyone who wasn't at Don's talk, I'm going to just really briefly kind of talk a little bit about sponge biology to get everyone kind of up to speed. These are really fascinating animals. It's a colony of these specialized animal cells, and they have the ability to filter an immense amount of water. They form this structure, and they do all sorts of crazy things with filtering and recycling nutrients. But one of the best ways that we can visualize this is by actually injecting a fluorescein dye into the sponge, which is what we were doing in our field activities earlier in the week. And this is a way to kind of visualize. It's hard, when you're in the water, it's hard to actually determine where the water's moving, right? Because we can't see it very well. So with the dye and some of these sponges, we can actually see how much water is physically moving through these sponges, and it's really quite incredible to witness. So, but I like to think about sponges as kind of this underappreciated, part of the reef, part of the marine habitat, and it's kind of like they form this infrastructure, almost like the plumbing in your house, right? Where it's sort of hidden, a lot of the pipes are behind the walls, and you may not pay that much attention to it, but when it stops working, you really notice it, right? It's like, oh man, something's going on here. So it's a part of this, these sponges are part of this ecosystem, and they play a really, really important role, but we tend not to really think that much about them. And so I hope that, Don really drove that point home and introduced us to the biology and the ecology of that. And I'll just recap some of that here. So one of the things that they do on the reef is that they filter an immense amount of water, as I mentioned. So they're removing phytoplankton, zooplankton, bacteria from the water. They're also engaged in this process called the sponge loop, where they're actually converting dissolved nutrients in the water column into particulate food for other things to eat. And it's a really amazing process, right? Imagine there's the ecosystem in the water, and the sponges are able to convert something that's dissolved into physical food for other things to eat. And in that way, it's able to recycle nutrients on the reef, which ends up being the basis of the food chain for a lot of other animals. So that's the nutrient cycling aspect there. Other ways that sponges form this physical infrastructure. It's habitat. It's three-dimensional space. Similar to corals. So up top there's a diagram of the morphology of different types of corals. We've got plating, branching, bouldering corals, and sponge morphology is quite similar, actually. You'll see a lot of sponges that have a lot of vertical relief, some that are encrusting, and even in some areas, so what we'll do as scientists, we'll go out and we'll try to measure the amount of structure that the sponges actually provide on a reef. And in some cases, the sponges will actually provide more relief and more habitat for fish and crabs and shrimp and other invertebrates than corals. And so that's a really kind of appreciated, I think, aspect of these sponges as well. And then so along with that, the sponges are increasing the productivity in these reef habitat. There's some really interesting studies that have been done where we can actually count the amount of fish in relation to the percent of the sponge cover. So in some of these reefs that are actually degraded in the Caribbean, if we have more sponges, you'll actually see more fish. And then vice versa, with less or fewer sponges, the fish abundance goes down. So these are really important parts of the ecosystem, and for things that we really like to see, like healthy fish populations, on the reef. And that's not true just in shallow habitat. We also see sponges in sponge nurseries in these deep water habitat. I think this is a study that was looking at these sponge habitat in Nova Scotia in 150 to 250 meters. And so really, really deep water. And we're finding that the sponges are actually providing habitat for fish nurseries. And they're using this habitat to find each other and to breed. And along with that, there's been some sponge declines, pretty typical of a lot of things that we're seeing in the ocean right now. And Don covered a lot of this, particularly the sponge declines in the Florida Bay. And I'll talk about this in a bit. But what happens, for this instance in the Florida Bay, what we saw with a lot of this disease and decline due to the cyanobacterial blooms, that was a lot of lobster habitat that actually just disappeared almost overnight, and that's a major commercial fishery for folks in Florida. So that was quite a large issue, not just in tropical shallow water areas. We've seen sponge declines in these colder, more temperate regions as well. So there's these amazing glass sponge gardens that are mostly off the coast of British Columbia, and these are actually calcifying sponges. And if you've ever seen photos of these things, it's just incredible. I mean, huge, hectares of these calcified sponges, and it forms this amazing reef that looks quite different than what we're used to seeing with the corals that are growing around here. But issues, of course, with fishing. Sometimes you have trawling, fishing boats that end up destroying this habitat. And then we can study these things, right? What happens? Oh, yeah, question.
So the glass sponge garden is a bad thing?
The glass sponge dying?
The glass sponge garden in British Columbia, is that a bad thing?
No, the glass sponge gardens it's a natural habitat. This is a healthy photo here.
Okay.
Yeah. Sorry, yeah, they're not quite as colorful. Maybe it's deeper water. They're not photosynthesizing, so they don't have some of these pigments like you might see on a shallow water sponge. But this is actually live calcified sponge, animals that form the basis for the structure. So this is what a healthy, good glass sponge garden would look like. And so we can actually, study how the fish communities change over time. Prior to and after destruction from these trawling fishing boats. And the blue line really is the rockfish. This is from a study from Francis et al., 2024. And before and after, I mean, this is a pretty stark observable difference in the amount of fish that were in this area, right? You destroy the sponge gardens in some of these habitats, and the fish just disappear, right? So on one hand, these declines are really depressing and to think about, right? But we're also able to learn from them, right? So what we've learned is, through studying what happens when these sponges decline, we can say, okay, well, what happens when we lose the structure, we lose the filtration, we lose some of these functions like the ability to cycle nutrients, and what is the result of that? I've mentioned fewer fish. The whole community structure can actually change. Right, so we see fewer invertebrates, fewer of these snails, crabs, shrimp, and really this can lead to an overall disruption of the food chain, and leading to these degraded ecosystems. And also recovery can be really quite slow, and we're finding that a lot of the damage is done occurs from human activities. So through the declines also, we've learned really that the sponges have these traits or roles in the ecosystem that have what we would say disproportionately large effect on the ecosystem processes, right, or the stability of the ecosystem. And so we consider sponges to be a foundation species. And so in the bottom left here, I'm kind of, trying to get a little creative with how I kind of visualize this. But imagine you've got an area of hard bottom with nothing on it, and then all of a sudden you have sponges. And then through that sponge structure that all of a sudden exists there. You'll have recruitment of fish, invertebrates, and this whole community ecology will kind of develop around these sponges. So they're foundational to ecosystems in some areas. And in other ways, we consider them to be ecosystem engineers. So in habitats where they're already existing, if they're converting nutrients from the water column to the surface, essentially they're engineering the ecosystem. So we consider them to be both of those two things there. So, okay, so now that we know, we sort of established how important these sponges are and what they do, and we have a general idea of, what do we do now? And thinking about this from the perspective of restoration ecology, or someone who's interested in trying to, fix some of the problems that we've really done to our ecosystems. Well, one thing we can think about doing is actually sponge farming. So there's this is kind of really interesting. There's a couple of ways to do this. This is really in the aquaculture realm of things. And interestingly, some sponges are farmed quite heavily for pharmaceutical purposes. So sponges produce these secondary metabolites, which I'll talk about a little bit later. Imagine you've got these sponges, and they're competing for space and habitat on the reef, and they're producing toxins, essentially, and engaging in this chemical warfare in order to kill the other sponge so that it can exist in the space. But as it turns out, some of these metabolites that the sponges are producing were able to learn what they are, synthesize them, and some of the chemicals are being used in cancer treatment and cancer research. And so this is just one example of things that we can really learn from nature. And so so sponge farming could be done for that purpose, right? And so there's some folks who are really learning how to do that. In some instances, the folks will hang these long ropes and actually hang sponges here on the left. Otherwise, these lantern nets are used, which are sometimes used like in scallop aquaculture for folks who have seen that. So there's a couple different ways to go about this. But in Florida, what we're actually doing is farming sponges for restoration. And this is really pretty interesting. So what we're doing here is using these bricks and chopping up sponges, which we did in our sponge dive, which I'll also mention in a bit. But this is a really cool process. So sponges are, they're not, they don't have these organs. This is colony of cells. And so most of them, what you can do is actually just chop like a section off of it. And then if you reattach it to a brick, it will end up recovering and then regrowing, attaching to this brick and then regrowing into a sponge. And some of these propagation methods are kind of borrowed from coral restoration, where you can do this asexual propagation, where you actually fragment a piece of coral and then reattach it to a structure, and then it regrows. Yep, Lynn?
Similar genetic concerns, like with corals?
Yeah, that's a good question. Yeah, so they do have genotypes as well, so that's a consideration. Yeah, so if you have a single colony and then essentially fragment it, you may want to introduce other genetics into that population if you're doing it for restoration, for example. Yeah, so that's a good question. Is that we are definitely need to be thinking about the genetic diversity of these sponges as well. But it's a good, this is a pretty good method that we're finding. And then after not a huge amount of time, maybe 2 years of growth for different species, you'll end up with this pretty interesting looking sponge garden. These are loggerhead sponges, which in the Florida Bay is the most abundant, or formerly most abundant, species of sponge. So this is really kind of monoculture, right? This is one species that exists here. And there's a lot of effort, especially after the sponge declines in Florida Bay, to reestablish the same numbers of sponges that were there beforehand. And this was just kind of a mad dash to put as many types of these sponges back into that ecosystem as possible. But what I really want to try to talk about now is the difference between farming and restoration. And I think that we'll find some analogies to Ken's talk with the agroforestry here, right? So we need to be thinking about all of these ecological traits and these functions of these sponges, and maybe not just trying to create monocultures. Same for farming anything for food on land, right? You might want to sometimes be thinking about your diversity of different types of plants. Yep.
So those sponges are the ones that you grew on the blocks?
Yeah,
So
exactly.
if they grew on the blocks, did they kept on growing on the block, and did they move the blocks somewhere else? Or they actually sort of pretty much rooted into place?
Yeah, so that's a great question. So these sponges here would probably be, sorry, the question is, thanks. Are these sponges like attached really well to the brick, and are you able to move them at this point, or are they attached to the substrate? Is that your question?
Yeah, and can you move them somewhere else?
And are you able to move them from here to somewhere else? Yeah, so at this stage, you can pick them up and move them. And so you might have a nursery, like you would have for horticulture. So you might want to grow them in one area from a certain size till they get big enough, and then move them from there to somewhere where you want to outplant them. And really similar to coral trees, right? So you might have one area where you're trying to produce this biomass of sponges, and then you want to export that to certain sites that you're interested in restoring. Yeah. But sometimes, yeah, if they do get too big and they're in a certain hard bottom habitat, and they can overgrow the brick, and then it would be a bit harder to move them. So that's a good question. You have to kind of think about the timing there. So where it kind of goes from farming into the restoration aspect, we need to think about what is restoration. And I think this is a good system to, sponges are a great thing to talk about, a great topic in that we can talk about restoration, but this really can apply to other systems, right? We can apply this to urchins, to corals, and we need to be thinking about sort of the same ecology, the same biological principles, and maybe even the same processes for how we go about doing these things. So, keep that in the back of your mind as we're kind of going through this, and how we might try to apply this to different species of sponges, or even different groups of taxa that you might see on the reef, like corals, or even other habitat. So really, the restoration is the science of recovering or enhancing the ecosystem function, right? And so that's what I would say is the distinction between farming and restoration is we're not just purely trying to produce this biomass, but we're actually trying to restore these functions. And we've already established what some of these functions of the sponges are, right? Filtration, the nutrient cycling, habitat. Sponges can enhance biodiversity. And the goal of the restoration really is to create these self-sustaining, integrated systems that mirror processes shaping the natural communities. And this can actually be really quite hard, right? When you think about how nature works, how many different factors, right? Light, all these, light, flow, food, these interactions between species, all of these, evolutionary processes that these animals have gone through over time, and we're essentially now trying to recreate that. And so we have to go back and try to learn about these processes in order to actually try to restore these functions that we're really interested in, right? So, and I realize that this might not be the right audience for this, but there might be some baseball. Yes, bets in the crowd. Go Dodgers or Yankees. So, yeah, right?
Yes.
So this is from Field of Dreams for folks who, I hope most have seen the movie. If not, great movie. And that's James Earl Jones, and he has this line, right? If you build it, they will come. And so this is kind of a theory in ecology also that, okay, what happens if we just replace whatever we're interested in restoring, right? Sponges, corals. What if we just put, a million sponges back out there? It doesn't matter what they are. Could be same species, could be monoculture. Would that, lead to the recovery of these functions and then lead to the healthy ecosystems that we're interested in having? Well, maybe not so much. It's really ends up being a little bit more complicated than that. So instead of random planting, right, we might want to start thinking about some of these ecological theories. And I will admit that a lot of folks will give us ecologists a hard time because we use a lot of jargony words, right? We use these terms: foundation species, ecosystem engineer, and they each mean really specific things. And it can sound kind of annoying, to be honest. Yeah, and we sound like we're just better than everybody else because we use all these big words. But what I want to try to impress is that there's sort of a reason for using these really specific words, and there's a method to the madness, right? So I'm going to try to just walk through some of these ecological theories, and kind of hope to convince you that they're important for considering how we undergo restoration and bringing really the theory into the practice. And this can be applied from sponges to corals to urchins to really anything. So one of the theories is that there's this structural diversity is really important in ecosystems, right? So if you're thinking about a flat surface on a reef or in a anywhere really, it could be deep water, and you've got no structure, right? Nothing's there. Fish have no place to hide. Okay, well then let's start building structure into there. But the complexity is really changes at different scales. So you might have, for instance, a branching type of sponge that works really well for this microrefugia for really small snails, crabs, and other small invertebrates or small fish, right? So that's important. Then you might have these large, barrel type morphologies, like a barrel sponge, right, which provides a huge amount of this vertical relief, less of the micro habitat for smaller things, but a lot of good vertical three-dimensional space for bigger fish, right? So that's important. So you might want both of those things. Then you might also have these big sphere or globular morphology sponges, and some of those are really good for holding the substrate and keeping the substrate sort of intact, right, for sediment stability. And so which one do we want to pick, right? Which one's more important than the other? Are they all important? Which ones do we want to focus on? And so these are kind of the questions that we need to be thinking about when we're going through this process. Also, different species have different roles. So some species are redundant, where you might, say you've got, $20,000 to restore a reef with sponges or corals or whatnot, and you have to, yeah, question, Beth.
Sorry, I didn't mean to interrupt.
That's good.
My question is, when you talk about the different structures
Yeah.
of sponges, when you do the cutting off and starting them up again, do they replicate what kind, whether they were a vertical or a small? Is there a difference in being able to replicate those?
So the question is, are we able to replicate the structure? Yeah.
From a barrel sponge
Uh-huh.
and zip tied it down,
Right.
or whatever, would it replicate the shape of a barrel sponge?
Yeah.
It
Yeah.
would? Okay.
So the question is, if we take a cutting or a propagule and we attach it to a brick, will it eventually regrow into the shape of the donor colony, or where the individual that you took it from? And the answer is yes. And so with enough time and enough growth, it would reform the structure of whatever you took it from. Does that answer your question?
Yes.
Cool. So yeah, so then the next thing, maybe we need to consider which species, right? In addition to the structure, different species have different roles. So some sponges may have the same structure, but they might have different roles on the reef. So some might be redundant. Some may have more or larger functional effects than others. And so maybe we want to choose, if we're trying to have, be efficient with our money, and we only have so much money to do these restoration projects, maybe we want to choose species that are non-redundant, or you'd have a bigger bang for your buck, essentially, or a bigger impact for an individual species, versus choosing ones that are redundant, where you might get the same type of functional effect from multiple species. So we have to think about that in addition, right? And then another theory is that species interact. I talked about this a little bit. You know, they've evolved these evolutionary adaptations over time to compete with each other in these habitat. And so some sponges will undergo direct warfare with each other. This is a barrel sponge on the left that's actually overgrowing a tube sponge. And so we might not want to pick both of those species and place them really close to each other, because then we've sort of wasted our money and restored a sponge that ended up being overgrown by another species. Otherwise, these sponges that have these produce these chemicals, maybe we don't want to choose those that will end up killing other sponges around it.
So, are those the anti-cancer chemicals you were talking about?
Some of these, yeah, some of these are, I believe, yeah.
Anti-proliferative. Makes
Right,
sense,
right, right.
Yeah.
So, like I said, you know, what we're really trying to do is marry the theory and the practice. So these are some of the ecological theories that we just kind of have to talk about. There's species succession. Same thing. This is thinking about Ken's talk, right, where he's planting trees that provide an initial shade for undergrowth, and then he'll get rid of those trees, right? And then as soon as you get enough growth from the tree that was in the undergrowth, then you remove the shade. And so there's this natural succession that you're trying to do in agroforests, right? And maybe the goal is to try to accomplish similar things when we're essentially farming for restoration in the ocean. So we're thinking about species succession, these interactions, how do they compete? Do they facilitate growth for each other? Does the diversity of shape and form matter? Is there disturbance functions? And then there's the practical restoration, right? What do we choose? How many do we put out there? How far do we put them apart? When do we put them out there, right? And so this is the practical aspect of it. And the science of restoration ecology and what we're doing is trying to link these two together so that the practical restoration can teach us about the ecology, and in turn the ecology can then help us to inform how do we go about doing the actual restoration. And so there's this feedback loop where we're constantly trying to do these experiments and tests for these things in order to keep learning, and over time, right, try to learn about how to actually go about this process of restoration. And sometimes it's really bad, right? So the results of uninformed restoration can be catastrophic. So this is a tire reef that somebody had a great idea off the coast of Florida in Fort Lauderdale to just dump a bunch of tires off the coast. Yeah, and the idea was that, okay, this is structure, right? Build it and they will come, and we just throw a bunch of debris out there, and then the thought was that it would end up attracting fish, and ended up turning into kind of a healthy ecosystem. But what ended up happening was that these tires, nothing was recruiting to them. The tires were leaching toxic chemicals. There was loose debris, so they were actually lifting up and getting carried over into other sites and knocking things away. And it's cost a huge amount of money afterwards to actually remediate that work, right? So it cost way more money to try to clean up these tires than it did to actually go and put the tires out there in the first place. And so this is a real problem. This is really what we want to avoid doing. And so we want to have informed restoration so that we're really trying to avoid this problem and not, at worst, we could make these things worse, these problems worse if we don't really think about them. And so some of this work has been done, and so there's really good foundational work that's been done in Florida Bay for sponge restoration. This is from a study published by Mark Butler, who I think has been in See and Learn program a couple times. He's a sponge ecologist. He's worked a ton in the Florida Bay. And this is one study that I think is just really, really cool and super interesting. And so what they did was they compared these sites, the A, B, and C. And so A is what we would consider a healthy or not degraded sponge site. B is a restored site, and C is a degraded site that was not restored. And essentially what this team found was that in the restored sites, some of the natural soundscapes started to return. right? Because there was micro refugia for these pistol shrimp and other invertebrates, small invertebrates that are producing a lot of sound on the reef. And the sound is actually a cue for recruitment. So when you've got that sound emanating from a reef, more fish will recruit there, and then you start to get the building blocks for a healthy ecosystem start to return. And so this is a really great example, I think, of sort of restoration in action, and a successful example of that. But really, unfortunately, where we're at with this sponge work in places like Florida, especially in the Florida Bay, is that there's this recurring, cyanobacterial blooms. And so it's a bit like, sort of throwing the sponge back into the fire, if you will. And so we can do this work, and we'll have sponges, and we'll restore them, and everything's great for a couple years, and then we'll get a big cyanobacterial bloom, and then it all kind of goes away. So we need to be thinking about that. And in restoration, at the end of the day, it's not the solution. It's not a silver bullet. We can't use this to fix all of our problems, right? We need to fix some of the source problems. Yep.
Is there any research done about creating sponges that are resistant to the algae?
Yeah, that's a really good question. The question is, is there research being done into sort of enhancing sponge resilience at a species level or an individual level to cyanobacterial blooms, right? Not that I'm aware of. It's a really good question. In Don's presentation, do you remember he showed that sort of pea soup with the cyanobacterial bloom? So that's just, the bacteria, the cyanobacteria creates this mucilage. It's like a slime, right? And so it just ends up coating everything. And so it would be hard to for any sponge species really to withstand that, if that makes sense. There are some, we're looking at, for coral restoration, for example, we're looking at the stress hardening and looking at certain genetic traits that might confer disease resistance or heat tolerance. And so that work might exist with sponges, but with certain other stressors like that cyanobacterial blooms, I don't think that's really possible. But that's a really good question. Yep, Lynn. Also a good question. Thanks. The question is, how far ahead is coral restoration from sponge restoration? And they're different systems. There's probably more work recently being done in coral restoration and more money in that space. But sponge restoration, I think, has been around for a bit longer due to declines in Europe, in like Greece, for example. So folks have been trying to work on that for a number of years. But coral reefs are really these, there's more attention there to these systems, and maybe for good reason as well. Yeah? True. Yeah, that's true. And maybe we can incorporate sponge restoration into reef restoration. And there's a number of ways to think about doing that as well. Yeah. But yeah, thanks for the question. So some of this work that's being done, especially in the Florida Bay, is sort of been stalled because of these recurrent cyanobacterial blooms. And so that's kind of where the next phase of our work comes into play. So now what we're really thinking about doing is selecting for areas to do this work in places where these cyanobacterial blooms don't exist. And so for us, that is in the nature coast of Florida. This is a really, really interesting area of Florida. It's sort of less populated. There's fewer people, but there's some really, really interesting nature there. That's why I guess that's why they call it the nature coast. And there's there's probably less pollution. There's a little bit less human influence in these areas. But also, it's really important that there's healthy sponge communities in this area. So we're thinking about this from an enhancement perspective instead of necessarily just a restoration perspective. Can we enhance sponges in this area and create more sponge gardens and sponge reef to build some of this ecological function in areas where maybe they'll persist over time? And so that's kind of our strategy moving forward with this. So a little bit about the habitat off the Nature Coast. It's really different from Saba. We've got a shelf here, West Gulf Shelf, that's really shallow. So we can go out 10 kilometers offshore and be in 3 meters of water. And so you have to really, yep, question? Is the area protected by government? Yeah. A sanctuary? Yeah. So the question is this, are parts of the Nature Coast coastal community protected, like as a marine park or protected preserve? There are some protections and regulations in place from both the state and the federal government. So we've got, for example, fishing regulations. So you can fish throughout most of this area, but you can only take a certain number of a certain species and a certain size. But there's really very few areas that are truly protected where you can't do anything, right? So and it is quite a large area, and there's not that many people. So if there was, I think it's really important to enact those types of protections, especially in areas where you've got, higher population, higher fishing pressure, and these things. Does that help to answer your question? Okay. Right. Yeah. Listen, you're preaching to the choir here. I agree with you 100%. Yeah. Unfortunately, in Florida, in the United States, we're a little bit more reactive. We have different problems. We have many problems. But I agree. It would be great to be certainly proactive about enacting some, no-take zones and some really strict enforcement of marine protected areas. But as historically, we don't really tend to do that until there's a problem, and that's kind of just human nature, right? So, but it would be great. I would love to see that. So this area is very, very shallow, very far distance offshore, where it stays very shallow. Most of the hard bottom community here is comprised of sponges and sort of non-reef building corals. So we've got low relief types of hard corals, different from what we've got in the tropics here, where we've got big strands of acropora that are really these big reef-forming structures. We don't have that in this area of Florida. So sponges are particularly important here because they comprise most of the structure and the vertical relief in this habitat. And in this space, there's over 60 different species of sponges that we find. And also, I love this picture. This is in Tarpon Springs. There's an artisanal sponge fishery, and I think Don also mentioned this. But in Greece, there was a while back a sponge decline, and a bunch of Greek sponge fishermen ended up moving to Tarpon Springs in Florida, and they were really harvesting these bath sponges. These are the ones that have the spongin and don't have any of the really sharp spicules, and they're great, super spongy. You can use them for literally for bathing. And for many years, this community of artisanal sponge fishers were harvesting these different species of sponges here. And with the advent of all these synthetic sponges, they don't really harvest them for sponges for that much anymore. It's more kind of you can go there and pay to watch the sponge divers go in the water, and it's kind of like sort of entertainment attraction now. But there's a history here, right? And so sponges are culturally important as well to the area. You have a question, Lynn?
But it didn't negatively impact the reef because they grow so quickly. Like
Yeah. So the cool thing about these sponges is that you can just lop, the top side of it. You can and leave the donor base, and it'll end up regrowing. And so when you've got communities of folks who are, passionate about their natural resources, they tend not to overharvest. And we didn't really see historical overharvest of sponges in this area. But they would like to see more sponges, right? So that's an area where we may not necessarily need the restoration, but we could enhance the sponges in this habitat and make everybody certainly more happy. So here's this framework. We've established sort of an area, and now we need to think about this process. And it's important that we go through this so that we don't have this uninformed restoration again. We don't dump a bunch of tires in the water again and have the same outcome. Yep. Question? The question is, what do they use the sponges for? Yeah. So essentially, before the invention of synthetic sponges that we use for cleaning dishes or cleaning, bathing, these sponges were harvested and dried and sold for home use, essentially. Yeah. So yeah, this is the framework, right? So we have to go through this process so we don't have these unintended consequences. One, we need to ask what the need is. What are we missing, right? What are we trying to enhance? We need to be thinking about that from the start. What species, right? Who can survive this? It's really a farming and aquaculture process that we're trying to convert into an ecological outcome, right? So not every species may be amenable to the fragmentation or the cutting process. Number 3, how do we farm these species better? This is a production question. How do we do it efficiently? We need to be able to scale this up. And then 4, we have to test these ecological theories. How do they coexist? How do they compete? Do they live in certain densities, or should they be spaced further apart? And then finally, after doing all of this, we really need to learn. Did I lose Mike? Hello? I'm back. We need to assess the outcomes, right? We need to ensure and verify that we're doing what we set out to do. All right. So in this area, in the Nature Coast, there's really a couple big commercial fisheries, and this is kind of where it gets into the fisheries aspect of the talk. So this is the Florida stone crab, really commercially relevant species. These use sponge habitat in that area. And so we, by enhancing sponges, we could really ramp up the commercial production of stone crab and turn it into a more sustainable fishery for these local communities, and that would be a huge plus. Also, shallow water grouper snapper fishery. These fish, these commercially relevant fish, are using these sponge structures for habitat in this area, and so if we can enhance these fisheries for the same reasons, that would be a huge benefit. And so let's move on to the second part of the process, selecting the viable species. So this is a map here of where we're really doing our field work here, and I'm going to walk through this project that we're working on as an example of this restoration or enhancement process. So here we are on the Nature Coast. This is in Hernando County, Florida. Pretty small population here. Those yellow dots on the map on the left, those are only a few kilometers offshore from this local community, which has really only maybe 800 people living there. So quite a small community, and they're all really excited for us to be doing this work there because they would love to be catching more fish and to be catching more crabs. Now, of course, we have over 60 species to choose from. And so we can't, we don't have the time, the money, the resources to go out and test each species. So we have to sort of make some executive decisions. Which ones do we start with? Well, we're going to start with the loggerhead sponge. This was the most historically abundant species in that area. We're going to go with this vase sponge. And the other four, the vase, purple, sheep's wool, and yellow, are actually sponges that artisanal sponge fishery in Tarpon Springs, Florida, was harvesting. And so we sort of had some understanding for that reason that we know sort of those are being harvested and not really overfished. So these might be amenable to this propagation process. And so we really started with those 5 species as sort of proof of concept. And the first, the next step was to set up a nursery. Right back to your question, right? So if we went out and found these donor colonies, harvested them, attached them to sponges, placed them all in one area, and then the idea was to grow this biomass from which we could then use the sponges for experimentation afterwards. So, but this is the first question here, right? So here's a picture of us. Again, we're probably in, 2.5 meters of water here, very shallow. And here's a picture of the nursery with different species, maybe 12 months or so after cutting and replacing them onto bricks. So we were, one of the nice things personally with working with sponges now, in addition to coral reef restoration, is that sponges, they survive better, which is really nice, right? Because when we're outplanting corals, we'll go back and then we'll have really poor survival. And a lot of folks here from the Conservation Foundation are well aware of this, right? It's sort of a really uphill battle. But when we come back after, was it a year and a half almost, and we see 85, 90, 97% survival in the nursery. So, okay, now we're getting somewhere. We're starting to identify these species that we think are going to do well or could do well within this process. And some that don't do as well, right? So the yellow sponge, 20% survival. So maybe we'll opt to, not use that species for restoration. And I love this picture. So just logistically, I hope folks are kind of interested about, how do we actually physically move sponges around sites, right? And we have to get creative as marine scientists. And this is a fold-up dog pool that I found at Walmart for, I think, like $30. And we blew it up, unfolded it on the back of the boat, hooked up a live well pump to it, and we just brought sponges up and kept them in there, and we were able to move them to different sites. And it ended up working really well. This is a colleague, Brittany Scharf, who was a extension outreach agent within that county. So she's got a lot of public sway there. So from our nursery results, we decided to move forward with the 4 species here: the loggerhead purple, sheep's wool, and vase, that did fairly well. And the next question, right? So we've got the species. We want to try to move forward. How do we improve the production? This is really the farming and the aquaculture aspect. We need to figure out, how do we grow these things, and how do we scale this up? So one of the issues, in addition to these cyanobacterial blooms with sponges, is hypoxia, or the lack of oxygen, especially on the sediment or the surface layer there. So you might have a scenario where these bricks, if they're really dug down into the substrate, they'll kind of interact or interface with that hypoxic zone. And it's essentially zero oxygen in the water, and that can kill the sponge. So we thought, okay, well, what if we just raise them up? Will that improve our outcomes? And then there's a couple other questions that go along with that. If we raise the sponges up, maybe they'll have access to more flow and access to more food, and maybe they'll do better. They'll survive better. They'll grow a bit better. So that was kind of our initial question. And you may be wondering, well, how do we actually quantify? How do we measure these things? It's a sponge. What do we do to understand how it's growing, how it's surviving? So we can actually take these height measurements, which is pretty simple, with a ruler, right? And then we measure the difference in the height over time. We'll take photographs from the top and then trace the outline, and then we're able to generate a distance, essentially, which is the circumference of the sponge. And so the same thing over time, we can tell through the height, how much it's growing vertically, how much it's growing laterally. And then we're also able to convert those measurements into a volume. So different sponges have different shapes, and we can use different formulas depending on the shape of the sponge to estimate a volume, so we can get a total volumetric growth for the sponge. And so far, these are some really sort of initial results. What we found really interestingly, and this is why doing these experiments is so important, because we really saw exact opposite of what we were expecting. And in instances where we were raising these sponges up off the bottom, we were actually seeing that it suppressed the vertical growth. And so they were not growing as tall as the sponges that were kept on the bottom. And that is really interesting to us, right? And so we're thinking, why? What's the thought here? And so far, one of the things that might be happening is that if you're raising them off the bottom, then they're sort of not competing as much with each other for food resources. And so if you're raising them, then all of a sudden they have more access to more food and more flow, and so they don't necessarily need to grow as tall as they would otherwise to be able to gain food. So that's an interesting result. One. The next question, so now we're getting kind of understanding how do we grow these things, what are some methods here. Now we're looking at the interactions between sponges themselves. So if we're taking sponges of the same species or different species and physically abutting them or putting them right next to each other, what is the result of that? Do they help each other grow? Do they prevent each other from growing? And so far what we found is that the sheep's wool sponge here, which is on this left bottom, doesn't play as nice with some of the other sponges. So with the purple sponge in particular, which is up top, it's really when they're paired with the sheep's wool, it suppresses the growth of the purple sponge. And so we're starting to, through this iterative process now, when we're going back and we're thinking about reestablishing more nurseries and producing more biomass, we might choose not to pair or place the sheep's wool sponge very close to the other species. So these are things that we're sort of learning along the way, and I do want to play the video, and we'll continue to learn from, right? And as we continue to let these experiments run, we may see more and more interesting results over time. The final point here, assessing the ecosystem outcomes, and we haven't truly been able to quantify this yet, because a lot of the work we've been doing is proof of concept. So, in science, we have to do a project, we get some money, and then we show that it worked, and then we use those results to apply for another grant and get some more money to continue the work. So we're really excited to move forward. We just got more funding, and now the objective is to really look at some of these ecological outcomes. Just by looking at the sponge nursery that we've set up, we're starting to see more stone crabs recruit here. We're seeing fish recruit. These are porgy. This is a type of southern pufferfish, and stone crab in the left there. So as soon as we have the resources to go back and actually quantify and measure these differences, we're pretty confident that we're going to show that we're actually somewhat successfully able to increase the biodiversity and species abundance of different types of animals on these sponge reefs. Now, this is also just a kind of a fun point here. This is our timeline for the project that we had started in 2024 after the nursery. These symbols here, I think we kind of know what those are. Yeah. So that's another big question, right, when we're setting up these nurseries. And for coral restoration, this is really pertinent, right? If you've got a restored coral site and you get a big hurricane that runs along here. That's okay. So hurricanes can be incredibly destructive, and they can ruin months and months and sometimes years of your work. And so this was actually last summer. We had 3 back-to-back hurricanes that came up into the Gulf, and you can imagine, after all this effort in setting up these nurseries and these experiments, and then I'm just sort of sweating bullets here, like watching these hurricanes come across our experimental site. So we had the first one here, that was Hurricane Debbie, which went just north. But you can imagine, this is just the epicenter of the storm. So we were really on the bands there. And I was watching there's weather buoys out offshore, that were submitting real-time data for wave height, and I think I saw at one point like a 25-meter wave, and this was of course offshore. But I'm thinking, okay, what does it look like inshore where our sponges are at? I was expecting just chaos. Then, of course, we had Hurricane Helene, a bit of a bigger storm, also went a little bit further north, and then Milton, also a big storm just south. And so we kind of like where the star is right here is where our site was. And you may think that we avoided a lot of the impacts here, but these storms are so big and there's so much energy here that there's really a pretty much quite a bit of destruction all up and down the nature coast. And like I said, I was really expecting just to come back and see nothing. Everything was gone. But to our surprise and joy, I will say, our experimental sites were pretty much still there. And the sponges looked fine. So this was a really nice outcome also, right? And so we're learning about the resilience of these systems as well. And we can kind of do some natural ecology studies by just observing what happens when a storm rolls through these areas. So that was really nice to see. And then I also like to kind of just present this here. And so you think, okay, Florida, it's pretty hot, climate change, temperatures are increasing. But then you're like, wow, what are these guys? This guy's wearing dry suits? Why, what's going on there? So this was in January of this year, and the water is so shallow that when we get a cold front move through, the water was down into the 50-degree Fahrenheit. So what's that in Celsius? That's it's cold.
Yeah.
It's 10 degrees Celsius or about there. And so, we went out there the first time. We're like, oh, it won't be too bad. And then we're starting to get like hypothermia. And a colleague of mine just wore a 3mm wetsuit. And I saw his eyes just like, going really wide and getting blue in the lips. So I said, no, we got to come back with dry suits. And this is a shot of us from the top side, from the boat. And so we're literally, 2 meters depth in 10 degrees Celsius water. So not all of the marine ecology is like fun and games. Sometimes we have to grin and bear it in really cold water. But I think that's it makes it interesting and more worthwhile as well. So more work to be done. We've got to do some more long-term monitoring. We need to do more experiments. We want to look at the diversity, different species, how we're placing them in different structures and different arrangements. We need to start really quantifying the ecological outcomes, right? The fish and invertebrate structure, how things are recruiting to these sponge reefs that we're creating, and what are the impacts on the water quality. And we're pretty excited about this work because we're, so far what we can tell is that things are progressing in a really positive direction for this project. But the big takeaways, sponges are this living natural infrastructure, and I think we should really be thinking about our natural resources this way, right? It's that these are this is infrastructure that's supporting these ecosystem services that we all really rely on at the end of the day. And so just like we have to go out and fix our plumbing and change the air filters in our HVAC, we need to be thinking about our natural infrastructure and how do we fix this up and make sure that it's working for us. And restoration and enhancement is really, this is a process and not a product. So there's quite a bit more money from sponsors and foundations and folks that are interested in restoration now that we're losing a lot of our ecosystems. And there's all of these ecological problems. So, we need to be thinking really intentionally about how do we go about this restoration process, and we need to be smart about it so that we don't end up with a bunch of tires just floating around off the coast of Florida causing havoc. And I hope that through this presentation, my hope was to show that we're learning through this process, and as researchers, we love just pure learning, for the sake of learning, but there's really also this applied aspect to it, where we can take the ecology and loop it back into the practical, and it's this iterative process where we're learning from our work over time. And yeah, the goal isn't just more sponges. It's not just more corals. It's not just more urchins. The goal at the end of the day is to recover the functions of a healthy coastal ecosystem, and that's really what we need to be focusing on. So, yeah, we've got a bunch of sponsors. We've National Fish and Wildlife Foundation, Florida Sea Grant, Coastal Conservation Association of Florida. This is an interesting group here who's funding some of this work, and they're just pure fishermen. I mean, they just love fishing, and so we told them, give us some money, and we'll give you more stone crabs. And it was like 5 seconds later, they wrote us a check. So there's ways to leverage folks who may not be as conservation oriented, but who at the end of the day, realize that we want the same thing, and so that's part of our job also is to work with folks and to engage them in a way that we can have mutual benefits. Yep. Yeah, so the question is effects of climate change and warming oceans on sponges in general. Yeah, it's a really good question. I don't really study that aspect so much. There are certain species that are more and less resilient to some of those stressors. For instance, the purple sponge, one of the sponges that we're using here, we find it on the reefs here. And so that is a species that we find it down 10 degrees centigrade up to average water temperatures here. So that's quite a resilient species. But then there are others that are less resilient to climate change, and I don't really have an answer specifically about that. I think it really depends on the species and the habitat. Yeah, but it's certainly something we need to be thinking about and considering. Question? Yeah.
Yes. So before, when you were doing the experiment with the elevation, you were saying that it didn't really work because they weren't growing in size because there was less competition for resources. But why is it necessarily that bigger sponges means healthier sponges?
Yeah, that's a good question. So referring to the experiment we ran where we raised sponges and we saw this opposite result as expected, where they actually weren't growing as tall as sponges that were sort of left on the bottom. And is that necessarily a bad thing, right? Is that your question? And so no, maybe it's not a really bad thing. It could be that some of those other metrics, like when we were comparing the volumetric growth of this same species between those two treatments, the volumetric growth was on average pretty much similar. So in some instances, they might be growing less tall, but increasing their lateral growth. And so we're looking at sort of all dimensions of the growth, but we're able to kind of decipher sort of response to certain changes of individual metrics. And it's not necessarily a bad thing. Yeah. That's also a really, really good question. Yeah.
What is CCA?
That's the Coastal Conservation Association. And that's the group of fishermen in Florida who are funding some of this work. And their interest is purely just more fish so they can catch them. Good for them, yeah. But like I said, if it's sort of there's this mutual benefit, right? Then I'm happy, and we're able to do some interesting science along the way. And where I'll leave the talk is taking these lessons abroad. Oh, sorry, Alan. No. I have to have the question now.
I was just wondering,
Yeah.
I actually wanted to start thinking for your talk, but if you're not finished.
This is the closing slide. Yeah.
So a great talk, but I was wondering, do the fishers when they're fishing for stone crab, are they damaging these functions with their thoughts or with their methods? Is there any tension there?
The question is when for the stone crab fishery specifically, yeah, they're setting out pots or traps, And does that damage any of the sponge, the natural sponge, in that habitat? I don't know, actually. That's a really good question. They're probably selecting for hard bottom habitat where you do find sponges. And So I would guess that, yeah, I mean, to a certain extent, there's probably affecting the sponge populations to some degree. I'm sorry? And corals, yeah. But there's, in this area, there's quite a lot of hard bottom habitat. And not all of it has sponge. And so I'm not aware of any studies that have really quantified impacts from that. Yeah, and I don't know if the fishing pressure is high enough to the point where you would see those impacts. Yeah.
That would be interesting, of course.
Yeah.
Restore the reef and we have more crab.
They just come through and destroy it. Yeah. And that might be a case where we want to, protect these areas, where we could have no-take zone or marine protected area where we're growing these sponge nurseries, so you could have spillover effects or something like this. Yeah. But that's a really good question. Yeah. Hmm? Runoff from the question is this runoff, freshwater runoff. I'm sorry. Sure. Is there runoff from the land, from agricultural production, or any type of industrial? Yeah. Golf courses. Yeah, we've got way too many golf courses in Florida. Is that impacting some of these sites? Yeah. Our site probably not, because there's really not that much industry around that area. And there's some freshwater inputs there, but the water quality is pretty good there. In general, certainly, absolutely. That is one of the big stressors that's affecting our coastal ecosystems in Florida. And in fact, we're looking at expanding this work down into further south into the Sanibel Captiva area, and much higher population, lots of golf courses, lots of folks with grass that they're fertilizing, lots of runoff, more industry, and yeah, in those areas, a lot of the hard bottom habitat has certainly been degraded. Yeah, and that there's big issues with red tide. If you've heard of red tide in that area. Yeah, and that's quite destructive, and actually has some human health implications. And I think David talked about this in his talk with the Carenia brevis. And so, actually, one proposal and one project that we're looking at is maybe trying to see if sponges are able to filter out those some of those harmful algal blooms, like the red tide. And so that's another project where we need to just start playing around and testing different species and seeing if they're actually able, if they have the capacity to actually remove some of those harmful algae from the water. But certainly, yeah, there's coastal pollution is a major problem. So I'll just finish with the last slide, and then I'd love to take some more questions if I have time. But so this is our field activity on, was it Wednesday, Tuesday, Wednesday, Thursday. So what we did was we maybe made some history. We established what I think is maybe the first sponge nursery or attempted sponge nursery in Ceiba. And so we selected a few species that we thought might be a little bit more amenable to being cut and propagated. And these are some cinder blocks that Alwyn and his team are using for sea urchin recruitment or settlement collectors. But there's no lines attached to them right now, so we kind of opportunistically asked if we could try to use those for establishing an initial sponge nursery. And so the idea here is, okay, well, we'll try it with a few different species. I think we selected 4 different species, cut a little section off each, and zip tied them to these cinder blocks, and if we come back and then see, maybe some will recover and actually grow. And of course, the question is like, what is the utility of this type of work around Ceiba or places like this? And maybe not as much as in Florida in these really shallow, hard bottom communities. But you might find that there's a function for these sponges here that might surprise you. I read one study about an encrusting sponge that was actually used to stabilize coral rubble. And this is a big thing that we're trying to understand, how to stabilize that bottom structure to allow for more corals to grow on top of it. And so you can use concrete or you can use some other synthetic things. But sponges might be a natural sort of solution for something like that. So in playing around and investigating with some of these in Ceiba, there might be a positive benefit to that down the line as well. Question? Yeah.
Where is this? Not this one, the photo before?
This one?
Yeah, is that Tigris?
This is at the ladder labyrinth. Yep. So we're excited to hear maybe how those do. And then I'll leave with this slide in honor of Don Berenger, who unfortunately is not here, but this is a photo of him doing a back dive off the boat. And unfortunately he had to leave a day early before we were able to hear this talk, but he's the PI or the lead investigator of a lot of this sponge work, which makes sense given his background in sponge ecology and biology. So anyways, I love this picture, and I hope he's watching somewhere. Thanks, Don. And with that, yeah, and if there's any more questions, I think I'd love to try to answer them.
Thank you, Aaron.
Yep. Oh,
Questions?
do we have time for like one more, or is that
Yeah.
it?
What's the deal with sponges? Like, do they thrive better in nutrient-poor environments, or do they use nutrients, or like how do they get their food, and like what food are they
Yeah.
growing on?
The question is, how do sponges acquire food? What are they eating, right? Do they use nutrients? Do they not use nutrients? So there's the answer is yes and no. There's, from Don's talk, there's high microbial community sponges and low microbial community sponges. So they have a lot of symbiotic microorganisms also that are within the sponges. Same as coral. Yeah, so in these high microbial sponges, they're actually, some of them are able to photosynthesize, and they've got a lower filtration rate. And so they're probably using more of like light and some of the dissolved nutrients for food. And then there's other sponges that are maybe lower microbial community sponges that have a higher filtration rate, and they're actually filtering out bacteria, picoplankton, zooplankton, and consuming physical particles.
It's not really the nitrates and the phosphates and like the stuff we put in agriculture, like as a fertilizer.
Yeah, some do use that. Yeah, but they're not quite as efficient as like fixing, or as sequestering some of those constituents as like seagrass or something, or the algae blooms. Yeah, so they've got this dual role depending on the species, whether they're recycling or retaining nutrients in an area, or actually physically removing particles and using those as a food source. Yeah, but they can do both. Yeah. Yeah. Right. Right.
Sometimes getting like a bunch of people that have the same age get together and bring school.
Mhm. Absolutely, yeah. The question is, do we have symposiums or like forums for brainstorming so different groups can collaborate and come up with new ideas and
And make sure you share your work with some
yeah,
people who have information that might be relevant
for sure, for sure. Yeah. So the main way that we do that is through scientific publications. So we'll write up our reports and publish them so anybody can have access to it and read it. But otherwise, yeah, there are symposiums, there's benthic ecology meetings, there's restoration meetings for sharing information. Yeah, that's a big part of science. Yeah.
Thank you, Aaron. If you guys are interested in asking more questions, he will stick around for dinner. We invite all of you to stick around for dinner. It's $15 for 2 different kinds of pasta. Thank you, Calibri Cafe, for sponsoring this event. And we will see you on Monday for our final presentation of the year. Thank you and have a good night.